Okay, today we're going to talk about enzymes. Enzymes are numerous proteins that are produced in living cells, and they accelerate or catalyze chemical reactions. So enzymes are a type of protein. Remember, proteins were one of our macromolecules, and they are going to accelerate, which means they're going to speed up a chemical reaction. What are enzymes? Most of them are proteins with a three-dimensional structure. Enzymes are required by chemical reactions in organisms such as digestion. If you imagine eating your lunch and then it sitting in your stomach, it would take a really long time to digest the food if you didn't have enzymes. So the enzymes speed up the rate of the reaction, in this case, digestion. Enzymes control the rate of metabolic or chemical reactions within your body. Metabolic reactions give energy to different processes that occur within your body. And this is just kind of giving you an idea of a three-dimensional structure for what an enzyme would look like. Okay, we've mentioned that enzymes help speed up chemical reactions. Well, we need to understand what a chemical reaction is. As we take a look, chemical reactions involve a rearrangement of the molecular or ionic structure of a substance, as opposed to a change in physical form or a nuclear reaction. When you look at a chemical reaction, chemical changes occur in chemical reactions. You start first with reactants. So with the beginning stuff are my reactants. In this case, I have the CH4, which is methane, and oxygen gas, O2. These two substances are going to react together. They're going to go and have a rearrangement of their structure. Notice how in this first one for the methane, you have orange is bonding with the green. The orange is representing carbon, green is representing hydrogen. And then we have our oxygens, which the atoms are being represented by blue. We're going to have a rearrangement that occurs on the other side. The other side is our product. So again, you start with reactants. Enough energy is taken in, the correct orientation, we end up with a new product. The substances that are forming on the other side of our yield sign, this arrow is known as a yield sign, these are the products. And notice how we have a rearrangement. That orange carbon is now bonded with the blue oxygen, forming carbon dioxide. The green hydrogens are also bonding with the blue oxygen, forming water. So that was the rearrangement. Our molecular structures we started with, the atoms rearranged and recombined with a different species, meaning a different atom, to give us a new substance, new products. This diagram is just showing you how you could have two different species, two different substances that are going to go through a chemical change. Again, these are the reactants. We get a product where they're bonded together. This is an example where they're bonded together and then they go through a chemical change. The product is going to break them apart. You could have a chemical reaction where you have a compound added with an element. Again, the rearrangement in those molecules, notice A and B for reactants combined with C. Over on my product side, A is now combined with C and B has been left out. And so this is the rearrangement that can occur. You need to know chemical reaction, a chemical change has taken place. There's a rearrangement in the molecular or ionic structures. What you start with are known as reactants, and what you end with are the products. Two major categories of chemical reactions. You have exothermic and endothermic. In an exothermic, think exothermic, exo like exiting, energy is going to be released, or energy is exiting, and the heat is given off as a result. So again, in our reaction, we would start with our reactants. We would get products, something new, but as a result of this reaction, you would have heat given off, and heat is a form of energy. Again, that is known as exothermic. Endothermic, think in, like something's going in, even though it's two different spellings. Energy is absorbed. The energy is going to be taken in, and it's going to feel colder. So again, you start with reactants. There's a chemical change taking place. We're still getting products, but energy is going to go in to the system, into the reaction. Again, that's known as endothermic, and it's going to feel cold. An example for endothermic, if you think of those little ice packs, the ones that are in first aid kits, and when you open them, they're at room temperature, but you can bend them and crack them. You release the chemicals inside, allowing a chemical reaction to occur, and then this will begin to get cold because it's taking in that energy, so it feels cold, and you could apply it to the injury to feel better. For a chemical reaction to occur, the atoms have to have the correct orientation and the correct amount of energy in order for something to take place. Chemical reactions doesn't just mean that different compounds or elements get thrown together and then something happens. They have to have the correct orientation and energy. If you look at our diagrams, each one of these circles, you'll notice they're different colors, meaning they're different atoms, and since they're different atoms, they're also a little bit different size. The white ones would all be the same. For the correct orientation, this atom, whatever it is, must collide right here where there's a bond. These lines in between are representing the bond. So the orientation is referring to where the collision occurs. This is where it would have to go to break that bond. When you look at this example, this atom is combining and hitting on the bond, but it's the wrong atom. And you guys don't have to get hung up on which atom's hitting correct and not. Just trying to get you to understand when you hear the correct orientation and energy, you understand what that means. So this atom is not the atom that would be one to break the bond. In this example, this third one, again, your collision is occurring here. That's the correct and incorrect, orientation. Again, has to be the little white atom hitting the bond. Here you have the white atom colliding to another white atom. That's not going to happen. So these would represent, these last three, incorrect orientation, incorrect spots where they collide. You also have to have enough energy. Just because this atom is going to hit where the bond is occurring, if there's not enough energy, then nothing happens. Imagine a set of Legos, and you guys know Legos snap together. They hook together like puzzle pieces. If you just picked up a group of Legos and threw them on the floor, you might have some that fall where the orientation is correct, but they're not going to land in such a way that they're already snapped together. You have to push those two Legos together in order for them to snap and connect. If you don't have enough energy, it's not going to happen. Same thing with breaking those Legos apart. You have to have enough energy in order to pull them apart. Just because you're tapping on where the bond or the connection of the Legos is occurring doesn't mean they're going to come apart. This second diagram is just showing you again with the atoms. Here where we have oxygen combining with oxygen, they're colliding. That's the wrong orientation, so nothing happens, no reaction. The second diagram, we have carbon colliding with the oxygen. That is the correct orientation, so that would cause a chemical change, provided there's enough energy. And in this example, there is enough energy. You have a rearrangement occurring for the elements. Okay, there are 5 ways to speed up a chemical reaction. Again, for a chemical reaction to occur, you have to have the correct orientation with enough energy. And you guys must know these 5 different ways to speed up a chemical reaction. The first is to increase the concentration, meaning more particles. In other words, more of your reactants together. Fewer reactants, there's fewer collisions. But if you have an increase in the concentration, that means you're adding in more particles, more reactants. That's going to increase the chances that they would collide together. Decreasing the volume. In other words, having a smaller space. You could have the same amount of particles, and if they're in a very large container, fewer collisions. But if you put them in a much smaller container, the particles are now going to be closer together, and therefore you're going to increase the chances that they would collide together. You could increase the temperature. When you increase the temperature, the particles are going to move faster. Think about putting water on the stove. If you allow an increase in temperature to occur, you could eventually boil that water. Particles are moving faster. So when you increase the temperature, there's more movement because the particles move faster, again increasing the chances of a collision. Breaking up clumps can increase the surface area. If you have large chunks of a compound and you put them together, the outer layers could react. But if you break them up into small, tiny pieces and then let them mix, you've increased the surface area that's being exposed, therefore again increasing the chances of more particles colliding. And then you could use a catalyst. A catalyst is going to lower the activation energy. A catalyst is a type of enzyme, which we will talk about that a little bit more detail. When we mean lower the activation energy, this is the energy that's needed to get a chemical reaction started. Remember, for a reaction to occur, not only the orientation, but enough energy. Well, a catalyst can lower the amount of energy that's needed. It's not part of the chemical equation that occurs. It's just going to allow it to happen faster. We will watch these little videos in class. They're real short little video clips to help reinforce these concepts. Looking at biochemical reactions, biochemical are the chemical reactions that are occurring in our body. And there's thousands of reactions that take place in our bodies every day. Important ones are going to involve a special type of protein called a catalyst, or in a living organism, they're called enzymes. A catalyst is a substance, again, that is going to speed up a chemical reaction. It's not consumed within the reaction. It is not part of it. It is not going to be part of a reactant or part of a product. It's just something that is going to help lower the temperature or pressure needed to get that reaction started. Both enzymes and catalysts affect the rate of a reaction without being consumed in the reactions. All known enzymes are catalysts, but not all catalysts are enzymes. Remember, special protein called a catalyst, if it's a living organism, those catalysts are known as enzymes. Again, this is a short little video clip that we will watch in class. When we look at enzymes, they act on the reactants called substrates. Remember, in a chemical reaction, reactants are what you begin with, and then what you end with are products. Since we are looking at chemical reactions within a living organism, those reactants are known as substrates. They are specific, meaning they work with a specific substrate. In other words, there are specific enzymes that will work with a specific substrate or reactant. They do not mix and match. They are reusable. They're not permanently changed in this process in the chemical reaction. That enzyme comes back out so that it can be reused for a further reaction occurring in the living organism. When we look at enzymes, this green structure is representing our enzyme. And then where the substrate, this would be our reactant or substrate, it's going to fit inside here. Notice they kind of fit like a puzzle, or a lock and key is what it's known as, a lock and key mechanism. The spot where it's going to allow for the substrate to connect is what's known as the active site. Remember, I said they are specific. So you have an enzyme. This is representing the enzyme lactase, and it's going to combine with the substrate lactose, which is a type of sugar that it's going to break down. If you look at this first diagram, this top pink lactase, again, that's our enzyme. This side is our reactants. This is our reactant or substrate. Notice how the shapes fit together. So the enzyme's going to allow for the substrate to combine like a lock and key mechanism, combines at that active site. When you come over here, this isn't the full product, the product's at the very end. You see that they've combined together like a lock and key or puzzle pieces. And then you get your product. Remember, products are something new. And what's happened is you started with lactose, a type of sugar. The product is going to be a glucose and a galactose, two different types of sugar. But my enzyme is exactly the same. That did not change. Again, your active site is where your substrate temporarily fits in. This is my active site. And then this is my substrate, and it's going to fit together like a lock and key fit together. Some enzymes are considered inhibitors, like a poison. They're going to block the active site. Many of them end with this ASE ending. Sugars end in OSE. Lactose is a sugar. OSE ending. Enzymes typically end in ASE. Like sucrase, lipase, protease. These are all different types of enzymes. Notice they all end in that ASE ending. This pink was representing our lactase. Oh, one more example was catalase. Amylase, again that ASE ending. This is the enzyme that's found in human saliva that's going to break down the starch known as amylose. Lactase breaks down lactose. amylose, that's a carbohydrate type of sugar. Lactose, also carbohydrate type of sugar. They end in that OSE ending. But then amylase and lactase end in the ASE. They are the enzymes that are going to help break down the sugars. Okay, I've referred to this lock and key mechanism. This is known as a lock and key model, and you need to be familiar with the lock and key model for enzymes. They use the lock and key analogy because the key you would use to open up your front door at your house would not work to open up the classroom. You have to have the correct key with the correct lock. And that's true for enzymes. You have to have the correct enzyme with the correct substrate. They have to fit together like a lock and key model. So as you take a look, you again have your enzyme. It's going to combine with your substrate, which is your reactant. The substrate will fit into the enzyme at the active site. And that's where we're going to have a chemical change occurring. When the substrate is combined with the enzyme, it's known as an enzyme substrate complex. After the reaction, you'll have something new, different product. The enzyme is not going to be changed. It is going to remain the same. It is simply there to help speed up the reaction. Remember, they're specific. You have to have a specific substrate with a specific enzyme. They only work together if they fit like a lock and key. This is showing you another example. These at the top, these are just kind of drawn to give us the idea of what they look like, how they work together. But this is more the globular shape for an enzyme, which would be more realistic. Here's your active site and then our substrate where they would lock together. This is showing you the enzyme substrate complex where we have that substrate combined with the enzyme. This again is just showing you how the lock and key mechanism goes together, where you have your enzyme. Here's the active site. And then our substrate, which is our reactant. Notice they're the same shape, so they would lock together. Here they are combined. That's our enzyme substrate complex. Something's happening to give us a new product. So when you come over onto the product side, notice it's now known as an enzyme product complex. How do you tell the difference between the enzyme product complex and enzyme substrate complex? The substance for your substrate looks different. You have something different over here, so you know that you're now looking at the product. And then they would release those products, again, leaving the enzyme behind. It's not going to change. It can go back and find another substrate to allow for this reaction to occur again. Taking a look at how the enzymes help lower that activation energy. Remember, with a chemical reaction, we have reactants. They're going to go through a change to give us products. Well, reactants have to take in enough energy, remember the orientation, and enough energy before a reaction can occur. This is energy increasing on our graph. So I have my reactants, and when I first look at this, let's look at the red graph line. This is without an enzyme. My starting point and my ending point are exactly the same. That is not going to change. What changes is the amount of energy that's needed. So if I do not have an enzyme, here's my reactants. The reactants have to take in energy, 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 and then the top of the graph, the top of this hill, they now have enough energy where the reaction can begin. And then as it is returning to the product, now my reaction is taking place, things are happening, and then you get your final product. If you have an enzyme, it's going to lower the activation needed, which means it doesn't need to take in as much energy. So the hill, if you imagine this is a hill on our graph, is much lower. It occurs faster. Again, it doesn't change the starting point or the ending point. It just didn't have to take in as much energy, so it was able to get to the starting point much faster. It lowers the activation energy. This, from the starting point of energy, if you come across from your reactants, that's your starting point of your energy, to the top of your graph line, that is what's known as your activation energy. That's how much energy needed to be taken into that reaction before things happen. And so with an enzyme, it occurs much faster, much lower hill, much smaller hill. Then I can start forming products. So again, starting point and ending point, still the same. It just was able to happen faster. There are things that can denature or destroy an enzyme, and denature is a word you guys need to be familiar with, meaning what is going to harm an enzyme. A change in pH. pH, remember, is that measure of hydrogen ions. An extreme temperature change. This is why a fever is so dangerous, because if your fever gets too high, it can affect the enzymes in your body. And then concentration is also going to play a role in there. If there's not enough enzymes, then the reaction is going to slow down. So when you say denature an enzyme, you're destroying the enzyme. The shape of that enzyme is going to change. If you have a change in pH or a change in temperature, enzymes have to work at a specific pH or a specific temperature range for each different enzyme. It's not the same pH range for every single enzyme or the same temperature range. But when you look at a specific enzyme, you have to know the specific pH range and the specific temperature range. If it's outside of that range, then it's going to denature the enzyme, meaning it's going to change the shape. It can unfold the enzyme, changing the shape. It will no longer fit together like a lock and key for the substrate. When we look at the graph, we're looking at how there's a narrow range of conditions. In this case, we're looking at the pH. If you're looking at the pepsin's found in the stomach. It likes to work at a pH of 2. Can go a little bit lower, a little bit higher, but not by much. It's a small range that enzyme likes to function. If it's in an environment where the pH is much lower or much higher, it's going to denature or destroy the enzyme. When we look at the salivary amylase's pH range is between a little lower than 6 and then a little higher than 8. Ultimately, it likes to be at a pH of 7. Again, you get outside this range, it's going to denature or destroy the enzyme. And then the arginase, it likes to work around a pH about, follow this down, about 9.5 pH. So if you get, again, outside the range of where the enzyme can work, they will destroy them. It changes the shape of the enzyme. And that's what the little diagram is showing you, where you have your substrate. Remember, your substrate's going to combine with the enzyme, and their specific meaning, you'll see how it fits together like that lock and key. Specific enzyme has to combine with a specific substrate. But in this case, if that enzyme is exposed to temperatures over 40 degrees Celsius, it's going to denature it. It's going to destroy it. It's going to change its shape. And notice how the enzyme has flattened out, indicating the change in shape. Our substrate is no longer going to fit correctly. Some facts about enzymes. They are produced naturally by all organisms, meaning bacteria, protists, fungi, plants, animals. They all have enzymes. They're globular in shape, meaning they have to have that correct shape in order to do their job. They have to work at a very specific set of conditions. If the conditions aren't right, they're going to denature, which means it's going to change their shape. They would no longer function. Without enzymes, our intestines would take weeks to digest our food. If you could imagine your lunch sitting in your intestines for weeks at a time before it was digested, those enzymes speed that process up. How they work and what they do, enzymes again are going to speed up the chemical reaction in the human body. They're essential for respiration, digesting food, muscle and nerve function, among thousands of other roles. Each cell in the human body contains thousands of enzymes.